Welcome to the Bioinformatics CRO podcast. I'm Grant Belgard, and today I'm joined by Dr. Diane Shao, an attending neurologist at Boston Children's Hospital and instructor of neurology at Harvard Medical School. Dr. Shao is a physician scientist whose work focuses on understanding the genetic causes of childhood neurodevelopmental conditions, including how newer single-cell approaches can help answer questions we couldn't address before. She's also an investor. Diane, welcome to the show.
Thank you so much, Grant. And it's just such an honor to be here and also reconnect with you after our long time friendship from college.
Indeed. So what's been most energizing for you lately in your work?
Well, something I really like to think about in my work is how to span across disciplines. So, based on your introduction, I think the listeners can understand I do some very fundamental research in genomics. I also really think about how that research applies to patient translation. I actually see the patients and have to involve sometimes not yet solid data in terms of making a then firm clinical decision. And then as an investor, thinking about how to assess that landscape. And so all of these, I would say, require a different vision and goal in mind. And so I think a lot about for any given application, what is my vision of translation or patient care or understanding fundamentals, et cetera, and how to generate the data, work with the data, apply that to really further that vision, kind of like big picture goals.
What's a question you're hearing more often now than you were a few years ago?
The question I'm hearing more and more people want to translate, from the time, a PhD student starts working in the lab to postdocs graduating, postdocs trying to think about what's next in their careers, wondering about industry versus academia. There is such a strong focus on translation, making that impact on humans versus, I think, 10 years ago, as I was still going through training, it was more, you were doing a PhD. A lot more students were thinking about an academic path, fundamental biology. And, I don't know if this shift is good or bad, but it certainly brings new questions to the table. And in the, way far past, academia had this idea that, going to industry is, maybe a sellout, you're not asking us interesting questions, but I think there is a growing realization that those questions are also extremely interesting, extremely impactful, and need really, really smart people to be involved.
What does a good week look like for you? What kinds of activities make you feel like you made progress?
Yeah, I do think stepping back and seeing where have things gone from maybe you get some data that is really uncertain and murky. And then this week it's, hey, we can draw one small conclusion from that. Or it's thinking about, hey, this data, which is, applied to fundamental biology, may be able to be reanalyzed in some small way to give us an insight on an actionable clinical impact. Or thinking about, could this have implications for which companies we think could really be really strong in their market spaces. And so even one small 1% insight, I think is a good success because it's building on that, 1% in all different directions that ultimately leads to where we're going.
So since you have this kind of dual role of physician scientist, how do you think about differentiating between this is something interesting and this is something actionable, right? Because for your patients in the real world, you have to make decisions now. You can't wait 5-10 years for something to maybe be firmed up. So how do you approach that?
Yeah, those are really, I think, questions that the field of, let's say, genetics is always constantly grappling with. So I'll just give you an example from my clinic from this week, just to make it a little more pertinent. So this week, I saw a case in my neurogenetics clinic at Boston Children's Hospital, a 2-year-old that has a condition called lissencephaly, which is their brains are smooth, and they had clinical sequencing that comes back with a rare variant that's homozygous, meaning it's in both the mother inherited and father inherited alleles of a particular gene. and records the variant as variant of uncertain significance, which means on a clinical lab basis, they cannot provide a diagnosis. And if you search the literature for this gene, there are exactly 4 patients reported worldwide that have other variants, not this exact variant in this new gene, but their features are all extremely similar to the given patient. And so, there is a practical matter of how many people need to exist in the world for you to have confidence that, the 5th variant, in mother and paternally inherited alleles of a gene is now diagnostic. And so that's a clinical lab question. And, on their end, they said, hey, we can't call this, disease causing. We have to give it this variant of a certain significance label. And then there's a whole other decision to be made on a clinical level. So when the patient comes to see me and I'm like, okay, the lab is not going to be able to change your classification, but, I can tell you, your brain looks almost exactly like the 4 other brains that are out there. Your kid is manifesting all the symptoms that those other 4 are. We have a very good sense that we should be worried about the things that those other patients have. So, if they have, for example, those other patients had problems with their eyes. And so I'm saying, hey, we got to check their eyes. We got to check their hearing. These are the things I can worry about now. And those are really practical clinical decision-making matters. And then there's a whole, interesting aspect of, well, what do we do in this gray zone? And, those are kind of boundary pushing now research questions. So, I then spoke with one of the residents who, was very excited about this, potentially new gene and this new presentation that we're seeing. And so they're saying, hey, can we write this up? I'm like, yeah, it would be great. And, it would be great if we found 10 other people so that we now have the statistical informatic, confidence to provide this diagnosis. We can then go back to the clinical lab, change their classification. We would change, therefore, the classification of other patients that come in with a similar presentation and new variants in that gene, which otherwise wouldn't be diagnostic. And so we can then go back to the research realm and really make a difference. And so I don't know if that kind of showcases how the different elements of clinical decision-making, gray zones in what is known in a diagnostic laboratory, and then what can be brought back into the research side are clear from my description.
Oh, that's great. What kinds of uncertainty are you most comfortable with, and which kinds do you work hardest to reduce?
Yeah. So in terms of, I guess we can talk about a couple different settings here. So maybe in the clinical setting that I, currently in this example, continuing on, the kinds of decisions that we can make are interventional. Does this child need therapy? That's a pretty certain yes, and I can probably give a good sense of how much therapy they need. Do I need certain screening and certain organ systems based on what I know? The answer is yes. And, the risk that I'm going to be wrong, or, even if they don't totally need it, that screening will be negative. Okay, those are tolerable risks. But other risks are not so tolerable. Like, for example, if I am wrong about the variant interpretation and the family is doing prenatal genetic testing for, embryo selection for their next child, that, at that level, I may stop at my confident assessment that this is absolutely the disease-causing gene until I have more of my, research statistical evidence that I'm going to gather with my resident, let's say. And so those are, various arenas where I may or may not be able to make a solid decision. And then, stepping back into the research space, the confidence in a research diagnosis is a little more clear, because on a research basis, you don't need to have an assessment of any patient with any variant in that gene that comes in. You just need to have a sense of, is that particular variant doing causing a functional change? And on a research basis, there's lots of other a lot of other modalities that can give us confidence. You can look at the RNA changes. You can see how that variant affects gene function structurally. You can do other types of statistical testing if you enroll patient cohorts within your cohort to do, for example, linkage analysis or other types of confidence building metrics. And so in different settings, there are different ways to really increase confidence in different types of interpretations.
How do you think about measuring success when outcomes can take years to show up?
Yeah, that's a great question. You're kind of thinking about as we, let's say, push forward a research agenda on a given genetic condition, what the success.
Well, or in the case of investment, right?
Yeah. Okay. Yeah. No, I think that's a great question. So, why don't we, jump to the investment for just a moment, so right now, for example, not all rare disease genes are good, even current targets for investment, to even embark on starting a company. There are only certain, you'll see this mentality where people will invest in only a handful, let's say, of rare diseases, that are broadly of interest. And partly it's because those are the diseases that we know the most about. There is a lot more research dollars, a lot more research interest. Maybe the patient advocacy groups have been really promoting or focused on getting a therapeutic out, and there's enough support interest that finally there's enough data and understanding so that initial startup can even be conceptualized for investors to be interested. And so not every, let's say, genetic condition in this moment in time is ready for research translation. And so pushing that, long-term envelope, from the fundamental discovery of a gene to when is it ready to be even considered a therapeutic target, to actually pushing out the company, to then now assessing that market landscape and seeing whether or not it's worth funding, et cetera, is a really, really long pipeline, as you're suggesting. And so in any given moment, there are many different people, from investigators pushing their visions and agendas to the NIH, pushing their research vision agenda, to the, business development people pushing their agendas, and the investors pushing their agendas. And I kind of really see the progress for each individual needs to be unique. Like as an investor, I am really interested in pushing the investments that we make into rare diseases more broadly, but that doesn't mean every rare disease that's presented to me with the potential therapeutic target is a good investment to make. And so a progress on an investment front means having, grasped, let's say, further the landscape of a particular genetic condition, grasp maybe the market space, what are the FDA regulations. Those are things are progress in an investment space. Versus on a research setting, I may be a little more agnostic to which disease I'm looking at and promoting. And that research space may be promoting, let's say, like progressing new techniques for gene discovery. It may be, figuring out how can I collaborate better with others. And so for people in any given phase of all of these different intersecting sectors, I think progress at the end of the day is very, very individual. And I hope that collectively across everyone, this will really push the boundaries of treatment for any disorders, rare or common.
So across all the domains in which you operate, what is your expectation for the impacts AI will have in the near term, right? So looking out over the next 1 to 2 years.
Yeah, I've after this conversation, I'd love to hear your thoughts on that too, Grant. But for me, I feel like AI has touched every aspect of both what I do and also how I assess both the research spaces I want to go into as well as investment spaces I am considering. At a high level, right now, I would describe my usage of AI as increases in efficiency. So increases in data sourcing, let's say to help me find relevant papers and subject matter and people and spaces, et cetera. I also feel it as efficiency in terms of helping me integrate, across, different, let's say, perspectives. I want, right now I have all this data that describes this biology. Now I want to, understand how to change this into a clinical risk assessment model, et cetera. These are kind of, I would still consider efficiency spaces. That being said, I know that the AI field really wants to do new discovery, pushing the envelope, idea creation from AI. I don't feel that it's there right now. And, I'm not really engaged in tool development to know, how close are we to that. I think that pushing efficiency and data interpretation, management, et cetera, is already a really, really large task. It takes off so much from my plate to be able to outsource a lot of those tasks to AI, for it to also hold that information for me, across, let's say these are the grants I'm writing and this is all the data that I need you to store for me. And as I resynthesize into a new grant with a slightly different focus, how can you help me shape that? And then so it lessens my work a lot. And I've found it tremendously beneficial. And to me, that's really important because it means I can leave my mind space, let's say, open to big vision problems. I can be the one leading idea generation, and then using AI to kind of create these spaces. So I would say that's my perspective. I think AI is transformative, but I don't feel like AI is transformative in the way of taking the place of human creativity and pushing the boundaries of, let's say, the unknowns in the world.
When you're designing a study, what decisions early on have you found most impact downstream data quality and interpretability?
Yeah. So, the types of study that I design most are in the realm of human genetics. So I do some human gene discovery research, for which I would say the pipelines for that are probably pretty well described. And then I also do single cell technology development for the purpose of understanding how mutations arise, and in particular, understanding the variation in the DNA within or between individual cells of an individual, what we call somatic mosaicism. I'm part of a pretty large consortium from the NIH called the NIH Somatic Mosaicism Across Human Tissues Network, where analogous to other large consortium efforts, one of the most notable ones being the human genome sequencing project back in the 2000s. The idea is that by characterizing the full intra-individual variability in genetics, that tool can be extremely useful across many, many different areas of biology and life sciences. And so for single cell technology development, that experimental design really affects the downstream. So for example, I have been working on understanding human brain development and the single cell copy number landscape. Copy number changes are structural changes in the DNA where whole areas of regions of chromosomes either get amplified or deleted or lost. And so to detect structural copy number changes uses fundamentally different techniques than detecting other types of variations, such as single nucleotide variation, where you're just changing, let's say, C to a G or a single nucleotide, and also uses totally different techniques than identifying, let's say, repeat expansions in single cells, which are also highly mosaic across an individual. And so the study design choice of tool becomes really critical to say, is it even possible to analyze my genetic change of interest? And that decision comes down to a matter of, what is the goal of the project, and also has some practical considerations of cost, and also has some technical considerations of, do I have the informatic support to analyze the type of variation I'm interested in.
How do you communicate uncertainty to different audiences, scientists, clinicians, families, leadership?
That's a great question. Depending on the audience, I try to do things differently. Most people do a lot better with the things that are certain than the things that are uncertain. I think that for me, always trying to portray first what we do clearly know can be really, really helpful to then give a framework to all the things that we still don't know or still exploring. So just to give a, concrete example of that, in my work in mosaicism, I really think that there will be totally new possibilities for genomic biomarkers or different possibilities for precision medicine that are related to the genetic landscape when we look across all the cells in the body. But of course, we're still in early days of that on a research basis. So I don't know if that's true. But what I do know is true is that we have, for example, in our brain, in our neurons, hundreds of single nucleotide variation per neuron, times 6 billion neurons in our brain by the time we are born. So that is a biological fact. And so I can hang on that certainty and share with people that certainty, and then describe, what I think we can do with that level of genomic data, and, just so the audience kind of understands where I'm going a little bit with this thought. Think about, for example, the difference between when the Human Genome Project first came to light and they sequenced one human versus what we can do with the genetics now that we're sequencing hundreds of thousands of humans, across different countries, across different disease modalities, et cetera. That type of data, while we don't know yet what will be revealed about our cells and tissues and how they all work together from a DNA perspective, I think is also inevitable to shift how we think about disease and how we think about diagnostic possibilities.
What is the current state of the evidence on the impact of mosaicism on clinically relevant phenotypes and the prevalence?
Yeah, that's a great question, Grant. So in certain diseases, it is fairly actually commonplace now to think about mosaicism. There are a number of disorders where it's pretty common to now look for mosaic genetic causes. So, for example, epilepsy. There's a subset of patients with epilepsy which will get surgical removal of the epileptic lesion, and often somatic mutations are found in those lesions. They follow particular biological pathway principles, and so those are pretty clear. Another realm, which is pretty common to think about now, is vascular disorders, so localized cavernous malformations. There's a pretty common precedent. Vascular disorders like Sturge-Weber syndrome, which is a capillary malformation over just one part of the body, now are pretty commonplace. So there are certain disorders where it is common to now think about somatic mutations as the primary cause. There are other disorders that is coming to light that even while they can have both causes in the germline and at a mosaic level, that many of those individuals actually are mosaics. And just because think about generating a human, how many cell divisions that you went through to generate this entire person from the time they were an egg and a sperm meeting each other to, the huge, 5 to 7 foot human being. There's just a lot of mosaicism to be had. And that causes disease. And sometimes they look like germline presentations, even if the person themselves are mixed genetically. And then there's a whole realm of things that we don't know, which is maybe part of a subject of research, but things like, there are diseases where certain cell types are lost. So, for example, in Hirschsprung's disease, a very particular neuronal cell is lost from the gut intestine. And so, to me, that's a high likelihood place that there is likely a somatic localized, cell-specific component, but when it's lost, how do we use genetics to actually determine what it was that was lost to begin with? So a lot of questions, but I hope that answers your question on the areas that we do currently know, which is, I would say, a tip of the iceberg.
So how do you think about future development of, precision medicine and so on in a mosaic condition?
Yeah, so I'm really excited about a couple different areas. One area is simply leveraging the power of essentially what I would describe as, let's say, a saturating mutagenesis experiment within an individual. So, thinking about what we've learned from human populations. So when we sequence, hundreds of thousands of people from human populations, we can see, hey, these genes never have a mutation, and the other genes have mutations that are just scattered all across the genome. And those genes that never have a mutation, they're actually important to humans in some way. There's a reason why we never have a mutation. Usually it's because either they were embryonic lethal or they affected reproductive fitness in some way. And so that's actually a huge part currently of gene discovery, to compare to population databases and say, hey, those areas are constrained. This may be an important disease gene. And so similarly, you can imagine that there are lots and lots of disorders which don't have a strong reproductive fitness component. Think about, cancer, for example, in old age. It's not necessarily going to be selected against the population level. Think about, eye conditions, like strabismus, where you're not really going to have a strong reproductive fitness signal, or autism even, nowadays, many people are getting diagnosed when they're, already lived full lives. And so while some forms of autism will have a reproductive fitness constraints, others will not. And so then the question to me starts to be, well, if we can now get information on genomic constraints, so which areas of the genome are really, really important just in a particular cell type, so like in neurons or, in the lung cells or in something like that, is that now new information on what genes are really critical for biology? And does that tell us something about disease? So that's one area I'm really excited about. You can also think about that the same way in terms of modulation. How do individual genetics, within a cell either drive a phenotype or selected against the phenotype? So for example, let's say, a person with a neurodegenerative disorder where their brain, some of their neurons will die, with age. Well, it's not that the neurons die uniformly. Some will die earlier, others will die later, and there's genetic variation between that. So can we leverage that to somehow understand, well, what is it, genetically about those individual cells that are surviving longer? And I think in the past, the view is just, it's stochastic, some are just going to die sooner, some are die later. And yes, probably it is stochastic, but stochastic doesn't necessarily mean random. Stochastic is a distribution that is related to some underlying biology. And so, these are open questions as to, the genetics that drives these stochastic processes. And so these are some of the areas I'm interested in, and I think they have really strong translational potential and also the therapeutic potential.
When did you first realize you wanted a career at the intersection of medicine and research?
Oh, this is a great question, Grant. I think it actually goes back to the college, our college days. When we were at Rice University, I started working for a PI at Rice who's now left that university, but he was my first significant research experience, and I realized he was kind of a remarkable person in that he was a trained astrophysicist who then became an HHMI investigator, which is a very prestigious award funded investigator who studied slime molds, dictyostelium. And then when I was in the lab, was going into human immunology and had created a compound to treat fibrosis, which is I was working on in the laboratory as he had one postdoc and I guess me, the undergraduate, working on this at the time. And then he turned that into a company that was sold for, $1.4 billion, ultimately for trials in fibrosis. And so that mindset of the fundamentals of science can be leveraged across astrophysics to slime molds to human immunology to translational medicine. I think already came to me maybe through this experience by osmosis, maybe as an undergraduate in this space. And I think that mindset really resonates with me, as in at the core, science is science. And those principles apply, no matter what realms you're looking into. And also as, scientists or people engaging with life sciences in any way that many people do, you also don't have to be limited to the one dimension that you're trained in, that all of these realms are possible. And so, for me, that is what also made me think doing a MD PhD career path would be one for me, because it was one where I got to see both the research perspective, the translational perspective, the clinical perspective, and then, in my past couple years have added this investment and market space perspective as well. And while some people feel that they're really disparate, and indeed, they're really tackling very different problems, at the core, if you go to core principles, there's a commonality.
What's something you intentionally didn't do or stopped doing that made your path a little easier?
Oh, that's interesting. Yeah, it does sound like I'm just accruing things, but to be honest, I drop things constantly. I actually think that's a critical part to maybe going back to your question on what drives, progress. Progress does mean constantly cutting out everything that is not leading to your vision of progress. So, even in, let's say, my work on single cell technology, I was, developing some technology, applying it to a number of different settings. And when I found one that seemed like it's particularly interesting, in terms of its, understanding of the biology and, that we could really gain traction with the tool and stuff, it meant I just dropped everything else. And I don't have any intention of picking them back up unless they further my vision in a given direction. And so I think that there is always this fear, like the sunk cost fear of like, oh, I've invested all this time, I gotta finish it, it's gotta be a thing. But I don't buy into that at all. So I actually constantly drop things along the way. And to me, that's a huge driver of success, because it means we focus on our energy on the things that go toward a vision.
If you could go back and give your earlier self one piece of advice, what would it be?
Oh, probably don't stress so much. I think that especially as a trainee or a student, it was so easy to worry about how things would unfold and try to control them. But honestly, simply because we didn't know, for example, writing a first paper, you don't actually know what it takes to even write a paper, or, what are all the steps? What are all the pitfalls? What is everything you wouldn't even need to think about? And so I think I spent a lot of time stressing and, let's say, strategizing and things like that. But the reality is you just got to do it, and then you'll learn from it. As in, nothing needs to be perfect that first time. And to allow for the learning process, you're going to get more out of that than trying to make it go a particular way each time.
I guess on that note, how do you avoid burnout?
Drop everything else that that you don't feel like doing. No, but in some ways, I really believe that burnout is a combination of what we're holding, all the different aspects that we're holding, and also how we feel about it. As in, if we're aligned, like if I'm holding a lot of things that I'm doing, and those are the things that get me up in the morning, I'm so excited about them, I can't wait to discuss them with people and share them with the world. That's not burnout. That's just me holding a lot of things that I like to do. But burnout is having a particular interest, but also feeling like I'm obligated to do all these things I don't want to do. I'm supposed to be finishing XYZ thing in this other realm that I've sunk all this time and effort in. And so to me, preventing burnout is pretty continual, like every few weeks renewal of, what is my actual vision? What is actually driving me? And am I doing the things aligned with that? Because if it's not, and you're doing that and having that conflict internally long-term, that's what burnout is. So yeah. So if you are aligned, then that will feel good. Everything will feel like fun and flow.
What's an effective way to build competence across disciplines?
Competence or confidence? Competence. Oh, that's a great question, Grant. The biggest thing is to not be afraid and to not be afraid to not know. There's no reason you would know. And I find that what people really orient around is a strong vision. So for example, maybe with my own interest in mosaicism and thinking about how that can push our boundaries in precision medicine. I work in child neurology. I work in pediatrics, brain development, et cetera. I'm very interested in maternal influences on childhood brain development, but that's not a space I know at all. Like I don't know a single OB. I don't know nearly anything about obstetrics or, what happened, like all the actual biological principles of pregnancy, et cetera. And so, as I delve into that space, that is a totally new, space for me. But, what I do orient around is, how important I think understanding this phenomenon is. And if I can share with people my vision, what I know in a very clear way, others are going to want to help me, and that will build my competence. As in, I don't go in pretending I know anything about these other spaces where I don't. And that's actually where true collaboration lives. It's not, we both know everything about the other's field. It's knowing exactly what do I know that's valuable, between us, and exactly what do you know that's valuable between us, and then putting those together. And, and competence is not always getting to know everything in a different space. Competence is sometimes being able to know where the gaps are and know how to ask questions and get help.
What mistakes do you see smart people make when they try to do interdisciplinary work?
Oh, that's a really good question, Grant. You're full of good questions. So one thing I do think is really important to recognize is that there's always a difference in culture, no matter what. Research culture, medical culture, even as I'm talking about neurology research versus like obstetrical research, there's a difference in culture. And if you are not recognizing that and respecting those cultures, it's just not going to work out. So, for example, on the biological space, samples are really critical. I work with postmortem tissues. Those are really important. And, PhD scientists are also really, interested in studying human tissues. But why do PhD scientists have a lot of trouble integrating with MDs? It's because they kind of speak different languages, right? It's the way they're talking about the samples is different. The PhDs are talking about the samples as a biological utility. The MDs are talking about them like the boy they took care of, for 10 years and then passed away for some reason. And so to understand that culture is critical. If you go to the MD and say, hey, I'm looking for samples for X, like they might say like, oh, okay, I have some. And then you're going to say something like, okay, well, I want to study, protein, it's A and B and how they interact and like, blah, blah, blah. The MD is not going to connect with that, right? So, thinking about, well, protein A could be a therapeutic if it interacts with protein B in this way is a much more viable start. And then, also thinking about, it's easy to start thinking about, okay, the doctor is just the one who's going to just be retrieving the sample and et cetera. And the minute you start reducing some other person's role to just a task-oriented sample retrieval role, you've totally lost the collaborative interdisciplinary engagement there. And so I think about these things a lot, and I encounter them constantly. For example, even in my example of, what do I do as a neurologist who wants to think about obstetrical tissue? Well, when I started, I'm used to paying $0 for my tissue because, I get them from biobanks. I get them from, patient groups that are really trying to get people to utilize the tissue for studies, et cetera. But obstetrical tissues are different. They pay a lot of people, healthy pregnant women, money to collect samples to be part of studies, et cetera. And so even engaging on cost, what is value? I was running the risk of devaluing all of their tissue simply because I'm used to paying $0 for my tissues. And so these are all cultural nuances between disciplines. The same way going to a different country, you really have to consider those cultural nuances. Understanding them is non-trivial. I do rely on, saying things like, hey, I don't know what the typical way things are done in your field is. This is what I'm used to. And having that humility up front allows people to also share with you their culture and being open to that, whatever that culture is, and not just judging it as unreasonable or, too hard just because that's not the culture you're used to.
What's a good habit you find most strongly compounds over time?
Oh, good habits. I find that I think this may be going to your burnout question, finding the things that are going to make you feel passionate and excited every day. And sometimes they're not always scientific questions. Like, for example, I find a good habit that I have is taking a break at 2:30 p.m. every day. Either that break could be, taking my 2:30 meeting and, asking the person if they'd rather, take a walk around and have a discussion instead of, sitting at a Zoom screen, or that break could be, meditating for 10 minutes by myself in a quiet space. And so I guess I'm just, I mean that as in, not to say that everyone needs to take a break at 2:30, but just if that is something that you need and will make you feel good about your day, that's something you need to do for yourself. Similarly, if there's a particular question you need to answer to feel excited, engaged in science, you just need to go down that route, regardless of if it's exactly the right time or if you have, 10 other things you need to finish first or whatever it is, because it's doing those things for you that is really gonna make everything worthwhile.
And where can our listeners follow your various threads of work?
Oh, that's a wonderful question. I am in the middle of building my own lab website, but for now, you can find me through the Boston Children's Hospital. I have a research page there. I'll provide the link for your notes, and then also my venture capital firm is at legacyventurecapital.com.
Well, Diane, thank you so much for joining us. It's been lovely.
Thank you so much, Grant. So lovely to be here.
The Bioinformatics CRO provides computational biology services to academics, research institutions, biotechs, and pharmaceutical companies on a flexible hourly basis. Visit our website, bioinformaticscro.com, to learn more.